Hello world, I'm Sanjay Malik and I love flow cytometry. So in this series of lectures on the flow cytometry, today I'll be talking about sorting by using a flow cytometry. Okay, so the flow cytometric sorting. I'll be talking about flow cytometric sorting, and this is part 5 of the talk. Okay. So far we have discussed what a flow cytometer can do for us, and in order to do what it does, what are the components required, how the different components work, and how data is generated. So these are the things that we have discussed so far. So we can identify the cells, or we can appreciate the cells, and we display the cells uh, on the charts and plots. Okay, so for example, this is an example where we have two fluorescence displayed and we have got three population. Okay, so we have got the population. Now the cells are coming over here. So this is what we are finding. Okay, so yes, so this is what we are finding. Okay, so this is what is the scenario we have. That means this is a sample where it's a heterogeneous population, where we have a population that is that is showing stain for one fluorochrome, or that is expressing one particular protein. The other cells, they are expressing the other kind of the protein, and then we have some cells which are not expressing either of the protein. So we have basically, so the sample here is heterogeneous sample. Now for my study, I want all of this population separated. I want to do my requirement is I want to do a downstream analysis of the transcriptomes. Okay, transcriptomes transcriptome analysis for all these three population, and I need them separated. However, my sample is heterogeneous and all the cells are mixed together. In order to separate these cells, there are other methods also that can be employed, but flow cytometry offers multiparametric analysis. So that way it is superior when we are using many parameters as compared to the other methods like magnetic sorting or antibody based panning and all these things. So how the sorting happens? By flow cytometry, we can distinguish. Okay, we can appreciate the different population. We can appreciate these three different population. Now, how I can get this population? So, in layman terms, I can say that I rely on the instrument. I do the gating. Okay, I can do the gating. So it is gate 1, this is gate 2, and this is gate 3. And I tell the instrument, okay, fine, sort the cells of G1. Okay, G2 and G3. Okay, so then the instrument can sort and separate to me. So that is how I'm relying on the instrument. But how does the instrument perform this sorting? Okay, how the instrument is performing this sorting, we'll talk about that. In order to sort, what it is required for the sorter. Okay, so we'll see a general structure or general anatomy of a sorter. Okay, so if we are talking about a sorter, so the sorter is something like this. Okay, so there is a flow cell. Okay, there is a flow cell, and the sample is injected into the flow cell. So there is somewhere a sample tube is there. There are cells in this. Okay, and the seed fluid entered into the flow cell, the sample entered into the flow cell. The same way both the seed and sample here are driven by hydrodynamic focusing and by hydrodynamic focusing, and we have the cells here, and there is a laser. Okay, there is laser illuminating the samples, and as the cells are crossing the laser, so as the cells are crossing the laser, it's illuminating the cells, and the signals are collected. Okay, be it forward scatter, side scatter, or fluorescence. So from this signal, what we see, this signal is displayed onto the computer and are displayed on our plots. We see these three population, considering these three population. That means anytime a cell of interest is crossing the ledger here, the cell show up here as a dot. The other type of cell, when it is crossing the ledger, it shows up here, so on and so forth. So we set the population. So what happens as the cells are crossing the ledger here, and the cells, they travel through and they go to the west. If you are not doing this, then there is a west cup. Okay, so it takes the sample into the west. So basically the cells, it forms a stream, and I'm talking about a stream in air sorter, stream in air kind of sorter today. It's called stream in air. Okay, so that means there's a stream is generated and it comes into the air. Okay, and most of the high-speed sorter are actually stream in air based sorter. There are other sorters there are which can physically pick the cell. Okay, so there is a actuator arm which can physically pick the cell. Now there are switch acoustic switching systems also there. So those are the different type of sorters available. I'll be primarily discussing about the stream in air sorter, and that is something going to be there for quite some time now. Okay, these are fast. And so here what is happening, the cells, as the cells are getting illuminated and then they go into the west. If I wish to collect the cells, separated, okay, so I have two population, I want to sort them separated. Okay, then how I can get it? If you look into the textbook, this is what we see in the textbook. Okay, so the cells getting separated into the two different compartment or two different tube, and the cells that is not sorted this is what I'm seeing in the textbook. So how it deflects, so how it does this. Okay, so let's look at the process now. Okay, this is a general anatomy. There are voltage plates. Okay, and this is how it happens. So let's dive deep into this component. So I started from the flow cell. Okay, so this is a flow cell. In the flow cell, the laser is illuminating the cells. Okay, so there is laser is illuminating the cells. Okay, so the laser is illuminating the cells. It could be like this. Okay, so as the cells are getting illuminated by the laser, there is light and fluorescent scatter happening that shows up on the plot. Okay, that shows up on the plot. But as the cell is illuminated, then it is coming in the plot. We make a decision. You have to first define. Okay, so we just detection first. We detect the cell. Yes, whether the cell is of my interest or not. Okay, whether the cell is of any other interest. Okay, so whether the cell is in my interest or not, where the cell is coming. So the moment it is detected, okay, and then what happens to the cell after the detection is done? The cell moves. Okay, the cell is not stagnant, it is flowing. Okay, so the cell moves, and then what we use is called a nozzle. Okay, so there are two types of stream in air sorter. One is cuvette based and the other is pure stream in air sorter. So here I am talking about a cuvette based sorter where the cells are illuminated in the flow cell cuvette. Okay, in the flow cell cuvette, and as the cell is exiting the flow cell cuvette, there is something called nozzle. Okay, so say for example we put it this way, nozzle. A nozzle is a device. Okay, and this a nozzle is a device which has there is a orifice in the nozzle, and the cells, they exit through the nozzle. Okay, and why we put the nozzle here? Okay, so here if we don't have anything, then it would be like a thick stream of sample and see through that is coming out of that. Okay, we won't be having control over that. We won't be having control over the stream. Okay, so there are two purposes with the nozzle that we can do. One is we have a control over the stream, and number two, the nozzle is used to vibrate. So there is a piezo actuator associated with the nozzle, and the nozzle can vibrate. Okay, so we use vibrational state of the nozzle. Okay, so the nozzle can vibrate. So why do we need to vibrate the nozzle at all? Okay, so if you don't use anything, if you have a fine orifice here, then also it will it will make a stream fine. So it makes a stream. Okay, so it makes a stream. It makes a stream, and the stream would break into droplet at some point of time. The streams wouldn't be continuous always, depending on the velocity of the stream. The stream would break into droplet at some point of time. However, this breaking is uncontrolled. If we are not using any energy, then this breaking of the stream is uncontrolled and we won't be achieving the desired breakpoint. Okay, so I'll talk about why you would need a desired breakpoint. But using a vibration, so if you use a vibration here, then by vibrating this, we can adjust there are two things, that is frequency. Okay, so how many times the nozzle is vibrating per unit time, and what is the amplitude. Okay, what is the extent the nozzle is vibrating to. Amplitude. Okay, so frequency and amplitude, that is adjusted for the nozzle vibration. Okay, so if we vibrate the nozzle precisely, we maintain the frequency and the amplitude, then we can control where the stream could break. Okay, so where the stream could break, that we can control. Okay, higher the frequency, okay, so higher the frequency, the more the number of droplets it generates per unit time. Okay, so it is vibrating faster, the more the number of droplets it generates, and lower the and when we want to break it at higher or lower position, that position of break off is governed by the amplitude. Okay, basically it's a combination of both frequency and amplitude that is desired to break the stream at certain point. Okay, a desired point, that's called drop break off. Okay, that stream break off. So by having a desired breakpoint, what is the advantage? So here, as the cell is getting illuminated, okay, and then it moves. Okay, then it moves. But as long as the stream is continuous, the cell is still in connection with the system. So the cell moves in the system. Okay, the cell moves in the system like this. Okay, and as the cell is reaching the edge of this, so here, since we are using a frequency and amplitude to control the break off, then this distance, okay, so the sample from detection to the edge of the stream, that remains fixed. Okay, so this remains fixed. Okay, and this is called drop delay. Okay, so this is called drop delay. Okay, that means how long it takes to break into drops. And once if we can control this break-off point, okay, we know the drop delay. There are processes to find out the drop delay, that I will be talking in a separate chapter altogether. So here I'm just talking about, say for example, this is fixed. Okay, this drop break-off point is fixed. We know the drop delay, and then what we can do is we use electrodes, okay, and charging to charge the stream. Okay, so how it happens is, so first is we have made a detection, then it will be deflection. How do we deflect the cells into the desired direction? Okay, so how do we deflect the cells into the desired direction? Okay, so if I want to collect the green cells, okay, the cells that is bound to the green fluorescent molecule, okay, then we have cells that are bound to the red fluorescent molecule. If I want to collect the red cell to the, say for example, to the right, and I want to collect it to the left, so it's not that green has to go to left always. I can reverse it depending on the how I select. Okay, so here I say I give a command to the instrument, sort, say for example, gate 1, sort gate 1 to left, and sort gate 2 to right. Okay, this is what the command I am giving, and we have the drop breakup point that is stabilized fixed, and now we know what is this. Then when we give a command, short command to the instrument, what the instrument would do, the moment it detects a cell, the moment it detects a cell, okay, the moment it detects the cell that is falling into a desired region, then it waits for the cell to travel to the edge of the stream. Okay, it is as the stream is about to break in the system, what it does, it applies a charge. So there are electrodes that is associated with the flow cell, and one electrode is providing positive charge, the other electrode providing negative charge. Okay, so what it can do is it can charge and this can discharge. Okay, and this can apply a further negative charge. Okay, so and then there are in the instrument there are two voltage plates again. Okay, so these are maintained at very high potential difference, somewhere around 10,000 volts. Okay, this is a very high potential difference. So for example, this is negative and this is positive. Okay, so positive plate and negative plate. So as the cells and we want to sort the green cell. So here consider here the green cell is the at the edge of the stream. The moment it is at the edge of the stream, in order to deflect this cell, so at the edge, that means what in the next step what it is happening, this cell is breaking off from the stream and it goes to the droplet. So on flow cytometer we never sort cells per se, we sort droplets that contains the cell of our choice. So here as the cell as the drop breakup is happening, the droplet carries the cell. But here what we are doing, the instrument is given the instrument is set so to sort or deflect the cell to the left. That means on the left there is negative voltage plate. So in order to deflect the cell to the left, what we need to have a positive. So that means the moment the cell is going to exit the stream, okay, as the stream is going to break off, here the instrument applies a positive charge to the stream, okay, and as the droplet breaks up, so it carries a positive charge. Okay, so the droplet there is now positive charge. So there is positive charge on the droplet, and on the under the influence of the external electromagnetic field, what happens? It's a positive charge carrying droplet, they deviate towards the negative voltage plate. So they deflect towards the negative voltage plate, and then they are collected separately. So those which say for example there is a green cell, okay, followed by a red cell. Okay, so then what will happen? The moment the red cell is also tracked. Okay, the moment the red cell is reaching the edge of the stream is neutralized. Okay, so and then applied opposite charge when the negative charge, because on the right we have positive charge plate, so when it applies the negative charge and as the cell is breaking off, that point of time it carries the negative charge, and the negative charge containing or negative charge surrounding droplets would deflect towards the right where there is a positive voltage plate. So this is how it is. And what if there is a cell that doesn't have either of the fluorochrome, it doesn't fall in either of these regions. So for example, it is coming in this region, okay, so it doesn't fall in, it's coming in this region. Then in such case, if we are not giving a short command, we don't wish to sort this cell, then in such case what will happen? The stream stays neutral. Okay, so it applies a positive charge, then neutralizes by negative charge, further applies a negative charge, then the second cell is deflected, then it applies a positive charge and the stream is neutral. And when the stream is neutral, that means the droplets that are generated are also not carrying any net charge, and they will not be deflected on the effect of the external voltage plate. So they would go to the west. Okay, so this is how the separation happening. Okay, so this is detection, then here it is deflection, then here it is collection. Okay, so there are few things that we need to know about the nozzle. Okay, so there are different type of nozzle. One of the nozzle that is on BD, so they call it the BD area nozzle. Okay, brief area nozzle, it looks something like this. So there is a, and this nozzle that comes with various different size. Okay, it could be like a 50 micron nozzle, 70 micron nozzle, 100 micron nozzle. Okay, 150 micron nozzle, 85 micron nozzle, like that. There are different type of different nozzles with different size of orifice. Okay, and these nozzles, depending on the size of the cells, these nozzles are used. Okay, so the ratio of the cell to the nozzle size is generally they consider 5 to something around 5 to 7 times. Okay, so if the cell, if I want to sort a cell of 20 microns, I would be using a 100 micron nozzle. If I want to sort a cell of say 5 micron, and then I would be using something 50 micron would be good enough for that. Okay? So this is how we set the nozzles, which nozzle to be used. And while we are using this nozzle, we have to keep another thing in mind. The flow cytometers, we change the seat pressure. So that means on an analyzer generally the seat pressure is constant, whereas on a sorter we can vary the seat pressure depending on the kind of nozzle that we are using. Generally lower nozzle will have a requirement of higher seat pressure, and higher nozzles would require a lower seat pressure. So this pressure also here it affects how the stream breakup is going to happen. If we have a high pressure with a large bore nozzle, then there would be it would be very difficult to obtain a stable stream breakup. And the same way, if the pressure is too low, then also will not be able to attain a stable stream breakup. So here this is as simple as this is in very short. And then how we compute the drop delay and how different instruments handle it, that I will be talking in a separate class altogether. And here what you need to just understand in a very simple way, as the cells are illuminated, the detection happens. After the detection, the decision is made whether to sort the cell, which direction it will be sorted, and then undesired cell that will. So deflection, and then finally we do a collection. So detection, decision, deflection, collection. Okay, so here these are the detection, decision, and deflection. Okay, detection, decision, deflection, and finally collection. Okay, and finally collection. This is how it is performed. Okay, so I will talk in detail about greater detail about how a sorting setup is done, all those things. However, the few things that you will need to keep in mind is the size of the cell, the size of the nozzle that is used. Okay, and so here this is a simple and the size of the cell that is used, how we prepare the cell. There is a nice single cell suspension. The better the cell suspension which prepared, the better the sorting is. If there is a large clump that is coming that could clog the nozzle, then it would again completely distort the sorting process. Okay, and so this is a pivot based sorter, and there are other instruments where there is a jet in air. Okay, so the illumination happens in the air. Here the illumination happens inside the flow cell. Okay, and that's for the sorting. Now I would elaborate on the sorting with respect to the setups, with respect to the troubleshooting in some other video. Okay, thanks for your patience, and thank you for being with me while I was talking about sorting.